Hi friends, welcome to this lecture on which is based on a research article published in 2013 titled An Ex-vivo gene therapy approach to treat muscular dystrophy using inducible pluripotent stem cells. As you can see, it is a paper involving stem cells and gene therapy to treat a genetic disease. Let's get in. So in this it is what we are going to discuss is Duchenne muscular dystrophy, or simply written as DMD, right? So it is a X-linked genetic disease, and it is a severe progressive muscular disease, in the sense people as they keep progressing in age, they will eventually become more and more, the symptoms worsen, and eventually they die by the time they reach about 30 years old or so. So when they are born, around 2 years, usually there are not so many of symptoms, but sometimes they may have to use their hands to get up. I think that is called as Gover's syndrome. And eventually the belly keeps protruding and the back keeps bending up like this around 8 years and so on. And you can also see that the calf muscles can become disproportionately excess, and eventually they become more crippled, and they have to be carried around through in a wheelchair. So basically it's a disease of the muscles, deformities, and they cannot the muscles are not powerful enough to perform an action. So what we can again, to have a look at it, it's not just the skeletal muscle that is actually affected, like in, say for example, skeletal muscles of the legs, and they lose a lot of muscle mass, and there is a lot of inflammation and fibrosis. There is diaphragm is also made of muscles, which becomes very weak, and as you know, the diaphragm is important for respiration, and then respiratory failure may also follow. Similarly, heart, also myocardium also is usually affected, because that is also a muscle, and heart failure would result. As you can see in this picture, with the biceps is somewhere something like this. Over the time it keeps degenerating or becoming weak, and that with age that keeps on weakening further and further, and eventually I mean, when multiple organs, in all of which the muscle is involved, and as you can see, when there is respiratory failure or heart failure, it leads to death. There are several medicines, something like steroids, and sometimes operation, and many surgery, many other things are there, but they are actually not cures. They usually prolong the life a little bit more. It is a X-linked disease. The mother has 2 X chromosomes, and the males have only 1 X chromosome. The gene responsible for this dystrophin, we will discuss more about it in the later slides. It is present on the X chromosome. So a man is likely to have only 1 copy of the gene, whereas the woman has 2 copies, because there are 2 X chromosomes. However, if a mother is a carrier, which means that in one case she is carrying, in one of it is a mutated one and the other one is fine, so she is a carrier, she will not be affected, because the functional gene will produce the required dystrophin, and therefore she remains asymptomatic, but she can carry. So there is a chance that the progeny of that there is a possibility that they both are normal, but there is also 50% chance that if it is a girl, she might become a carrier, or if it is a man, if it is a boy, the boy may be affected, because as I said earlier, the female has 2 copies. Even if one functional copy of the gene is present, then it is all right. Otherwise, such as in the case of the boy, who may have inherited the default or the faulty or mutated gene, that person will be affected and will have to have a difficult life. So if we go further, we will try to understand the molecular basis of DMD, and for that we should understand that the whole muscle is something like this, which has bundles of muscle fibers, and each bundle of muscle fiber, they contain muscle fiber, and within the muscle fiber you find there is the muscle fiber membrane, that is this one, and there is actin filaments within this muscle fiber, and there is a connection between the several proteins that are present on the cell membrane. We will see it we will see shortly in the coming slides. Until then, I will just take it as this. Dystrophin is a protein which keeps a connection between the cell membrane or muscle fiber membrane and the actin filaments of the muscle fiber, and that is how it plays a function of keeping the muscle intact. As you can now see that here there are several types of proteins. I don't need you to mug it up, but maybe you should try to know about alpha dystroglycan, and there is beta dystroglycan. This is the sarcolemma, and here this part belongs to the muscle fiber. This is the sarcolemma, that is the cover, which will then keep it again through laminin 2. It will keep in contact with the basal lamina. So this is dystrophin. Dystrophin has 3 domains. One of it is bound to the actin domain, the other one is bound to this the proteins that are in the beta dystroglycan. Here it is bound to the beta dystroglycan, and it also has a rod shaped structure like this. So dystrophin is making the contact, and which is important for the integrity of the muscle fiber. There are multiple isoforms of dystrophin, okay, in different types of cells, or functionally different cells have different types of dystrophin synthesized. So we are not going into too much of details about it, but it's a good idea to remember that there are, in different cells, there different isoforms have a function, and that is how you should try to remember. But most often what we are discussing is this one, the one that is involved in muscle and other things. Okay, what happens in DMD, or there are also other dystrophy kind of diseases, that is called as Becker's muscle dystrophy, and here is the one that we are actually trying to study, Duchenne muscle dystrophy. So it is a gene, the gene is about 2.6 MB, that's a huge gene. It has about 97 exons. So I mean, I hope you can understand that it is the length of the gene in that sense. In Becker's, there is a large deletion of several reading frames, several exons, but the reading frame tends to be same, so that it actually produces a protein, but there is a large deletion in it. In DMD, however, the problem is that there is a deletion is very small, but because of the deletion there is a shift in the reading frame. So the say until this part the protein is as normal, but from the location of the deletion it has changed. It is no longer, and the sequence is no longer similar to that of has any similarity with that of the dystrophin. So from here on it is actually non-functional. It doesn't serve the purpose, and I think there is a premature stop codon that comes up somewhere in the reading frame, and because of which there is a shorter version of it. So we'll have a quick look about what happens because of this mutation. So quickly, as we already discussed, here is the video from which I have used. You can try to have a look at it. It's well illustrated. So here is the myocyte membrane. This is the extracellular side, and we have, this is the intracellular. Myocyte is kind of used many multiple cells actually. I hope you remember that. So here are the actin filaments that are shown in blue. The red, this is the dystrophin that is being shown. It has three parts. One is actin binding end. There is a central rod, the long stalk like thing, and then there is a the third part is dystroglycan, beta dystroglycan binding end. Okay, so that is how the connection is complete. Extracellular matrix or basal lamina to the dystroglycan complex to the dystrophin, through dystrophin to the actin filaments. So there is much more scope for integrity. If because of any mutation or something like DMD, because of a frameshift mutation, there is a truncated or mutated dystrophin like this, which does not have the dystroglycan binding domain, and therefore it is no longer in connection with this protein complex. Then each time the muscle contracts and relaxes, there is no connection between what is happening to the myocyte membrane and inside the myocyte. That means there is a larger scope for tearing of the myocyte membrane. So the myocyte membrane, somewhere, something like this, it gets disrupted, and if you can remember, there is always a polarity kind of thing maintained. The outside environment, the extracellular environment, is different from that of the intracellular, or across the membrane there is some kind of polarity maintained. Say for example, you will have calcium ions, high concentration of calcium ions on the extracellular side compared to that of intracellular. In the intracellular there may be, but it's usually in the smooth endoplasmic reticulum, right? So calcium, if there is any, it is usually stored in the smooth endoplasmic reticulum. So the cytoplasm usually maintains low calcium ions, low calcium ion concentration. But because there is a disruption in the membranes because of lack of integrity, or which is because again of mutated dystrophin, there is a leakage of calcium from the extracellular side to the intracellular side. So once the calcium ions that were present outside, they come in here into the cytoplasm, they activate multiple processes, which also include some of the proteases. These proteases are typically used to degrade any ubiquitinated or highly damaged proteins. But now this is abnormal activation, excess activation of proteases because of excess or high concentration of calcium, intracellular calcium ions. These degrade many proteins and eventually will lead to the death of the myocyte. There is also other problem that creatine kinase, which is important to maintain a creatine phosphate as a energy store within the muscle cell, that is the readily usable. Apart from ATP, creatine phosphate is another way of how how energy is stored for ready usage. Now, because the membrane is broken up, creatine kinase is released out or leaks into the blood, and then it keeps circulating. One of the ways of diagnosis of DMD is also trying to estimate the creatine kinase within the blood, circulating creatine kinase. So eventually, I hope you understand that if there is no energy in the form of creatine phosphate, because there is no creatine kinase, the muscle will not be able to function as well. So both these cases, increased activity of proteases like we have discussed little earlier, and also reduction in the creatine kinase would result in the death of the myocyte. So each time the myocyte dies out, say these are muscle fibers, and say these are the long bundles of muscle fibers, and some of them die out, those areas are filled up with fat or scar tissue, and they eventually interfere in the function of the muscle itself. So you can understand that through with age or with time, more and more myocytes will die out, and then that is how the skeletal muscles become weakened. So apart from many others, we will just quickly have a look at it. They have the back muscles are weak, because of which the spine becomes curved like this, something like that. And then the belly muscles are weak, so the belly falls up in the front. As we discussed, the calf muscles are excessively swollen because of fat deposition or a scar tissue formation within the skeletal muscle cells. They use the hand, they keep the hands behind to balance out or even out the belly weight in the forward. So these are some of the early symptoms. Actually, Grover's syndrome, when the baby is sitting maybe around 3 years or so, to get up, they cannot, because the leg muscles are very weak. They have to use the support of their hands to get up. They cannot get up just by their legs, because the leg muscles are not so strong in these situations. So there is another disease, another protein apart from dystrophin. It's called as utrophin. So which seem to have very similar in structure. I think in the next slide you will be able to better see it. This is the actin binding domain. This is the dystrophin, beta dystrophin binding domain, and then these are the like structures that are present in dystrophin. When mutations happen, they might result in smaller versions like this, and there is one protein that is called as utrophin, which is normal. A deletion of which doesn't seem to have a lot of a problem, but it's not X-linked. By the way, it is present on the normal, I think it's chromosome number 6. So why are we talking about utrophin? Is utrophin, so here is a quick comparison of dystrophin, utrophin. You can see that there is a lot of similarity in their structure. And if now the question is, an individual is there and the individual has a problem that the individual is unable to produce dystrophin. So if we want to make any kind of curative or therapeutic activity, we should try to replace the faulty gene with a good gene. But the gene is huge, as I was telling you a little while ago, and then a replacement can also be used as utrophin. But people have figured out, I think through trial and error, that they designed something called as micro utrophin, where it has the actin binding domains and some other domains, and this whole part is missing, and they made the rest of it. So all we need is something like this, right? Actin binding domain, we need the dystrophin binding, beta, the other side of the binding protein, and then this rod like thing, so that the connection is made between the actin filament and the membrane bound domains. So they have designed this micro utrophin as a, hopefully, as, because we want to reduce the size so that we should be able to put the gene into it. The delivery of the gene, if you remember from your genetic engineering or in the previous classes probably, the larger the gene, the more difficult it gets to put in it. So if we had a smaller version, then it will be nicer. So that is where this micro utrophin comes in. And now we want a vector to move, or we want a way of trying to put the gene into. We decided that we are going to use micro utrophin, which seem to be functionally equal and sufficient enough to that of dystrophin. So the chosen one is called as transposon based on Sleeping Beauty. It's a nice kind of name, Sleeping Beauty transposase. They called it Sleeping Beauty because evolutionarily it has accumulated a lot of mutations in fish genomes, and I think we all have a lot of genome transposes that are mutated, and thereby they are no longer moving around. So here are some of the one type. It is a TC1 like transposase in fish. They have taken different domains from different. So the whole the whole transposase has something like this, several domains, and all these marks are mutations. In different types of fish, there are different types of mutants. Domains are mutated, so they try to bring in all the domains together, something like this, and actually they have you can call it as revived the transposon, and that's why it was sleeping until now, so it is called as Sleeping Beauty transposase. So it has the if you look at the transposon, what is functional right now, we have there are inverted repeats, as would have any other transposon. The ends are very important for the mobility of, or for the movement of transposons. I hope you can recollect that you have two types, cut paste and copy paste. This particular Sleeping Beauty transposase or transposon moves through cut paste, so it gets, we will see it in the next slide too, it will get cut here, and for the cut to be made, the transposase should be able to recognize some ends. Those are usually the inverted repeats. As you can see here, IR, terminal inverted repeat. In some cases you might find it as called as TIR. And there are also DR, which is the actual domains that are recognized by the recombinase. And here is the transposase, about 360 amino acids. Here is the DNA recognition, that is, this is the gene. When it encodes, it produces a domain of DNA recognition, that is recognition of these regions, the DR. And then there are catalytic domains that makes the nucleophilic attack, and so on. Okay, so we'll see the mechanism of movement. We can use these Sleeping Beauty transposons for multiple purposes. Simplest is to you just have to put the inverted repeats and the genetic cargo, whichever you want to move across. and the transposase is usually expressed from some other location, so that the transposase is expression is also transient, because we don't want continuous expression. If it is the case, it will integrate in one place and again maybe remove from here and put it somewhere else. Then we don't have any control of what is happening. So we can do gene transfer by putting the genes here, and it can get integrated, and then if we had a promoter, there will be an expression of the transgene. Or you can also, it might also result in insertional inactivation or insertional mutagenesis. And we can try to do it in germline or in somatic cells. That is also, many things can be done. So here is the mechanism of how it is used in general. There are two vectors. One is the vector containing the transposon itself. The other one is contains the transposase. In this one, the organization, these are terminal inverted repeats, and this is the and in between them we have to put our gene of interest. Once the transposase is expressed, the transposase gene is expressed, transposases are produced, which interact or bind to the terminal inverted repeat, something like this. They make a cut and then they bind to the target, and the target usually is TA. Okay, when you have dinucleotides like this, TA, they recognize that, they cut here and here, and then they integrate the DNA. So on both the sides there will be a target site duplication of TA like this. Yes, that is the Sleeping Beauty transposon system. So now we will see what the authors of this paper actually have seen. We understood what is the biology of DMD. We understood the nature of dystrophin, function of the dystrophin gene. We understood about utrophin, and the other one also is we have a construct, that is micro-utrophin, a gene, a variant of utrophin which can be used to supplement or replace the function of the mutated dystrophin. And now we also have learnt about a integrating system that is based on Sleeping Beauty transposons. So we'll see what the authors have done here. So the authors, this is the same, inverted repeats and inverted repeats. Their function is, they are essential for the recognition by the Sleeping Beauty transposase, that is SB is a Sleeping Beauty transposase. Then they have put in a promoter, and there is a kind of epitope for finding out, for locating where it is, and then they have put in the micro-utrophin gene, and then there is a IRES, that is internal ribosome entry site. I think you should be able to recollect that eukaryotic mRNAs are monocistronic. That means they have one start codon, say ATG or AUG, and then several codons and TGA. One mRNA produces only one mature mRNA produces only one protein. But if you want one mRNA to produce two different proteins, in the last sometime there is a paper where we have learnt about self-cleaving peptides, but this is a different one, where they have an open reading frame which is a start and UGA say stop, and there is another open reading frame they have put for GFP, that is green fluorescent protein. But if the ribosome typically would recognize, EIF4 4E would recognize the G cap and then initiate or allows the formation of initiation complex, a ribosomal initiation complex. And so it is essential to have this G cap for translational initiation. But if there is a different second open reading frame, such as here designed, then you want the ribosome to somehow be able to make a initiation complex. For that, there are some elements, genetic elements called as IRES, that is internal ribosome entry site, and ribosomes can be, initiation complex can be organized here and produce GFP. So this one will produce micro utrophin, and from GFP gene will produce one GFP protein. GFP is for green fluorescent. It is nice to know when it is expressed and track our cells. If you want to see the cells are expressing, if they are expressing GFP, that means that cell must have had micro utrophin as well, right? Yeah. And we should remember that these people have done many other things, but we are not going into all the details. We will see the essential parts of it. So I hope you know what is meant by ex vivo gene therapy. Gene therapy comes in two ways. One is in vivo and the other one is ex vivo. If there is an individual with a problem, we give the viral vector with the gene of interest, and then this person, the gene is taken up by the cells within his body and then they get repaired. That is in vivo gene therapy. Ex vivo gene therapy is something like we take out the damaged cells and we repair the cells and then make more and then put them back into the individual. Where is the genetic recombination happening, or where is the gene therapy happening? Is it happening within the body or outside the body? If it is within the body, then you call it as in vivo gene therapy. If the gene therapy or recombination is happening outside the body, like as we have shown here, then we call it as ex vivo gene therapy. So the others are going to follow the same that kind of approach, where they have taken a double knockout mouse, in the sense a double knockout. It is knocked out for dystrophin as well as for utrophin. So that both the genes are there. In cases where dystrophin alone is deleted, knocked out, the utrophin seemed to be taking its place, and it's not showing as much as phenotype like as in the DMD should. So they made a double, they took a double knockout which is deleted for both dystrophin as well as utrophin, and from there they have taken the tail tip fibroblasts from that mouse, and then they have reprogrammed to make induced pluripotent stem cells. Induced pluripotent means they can give rise to any kind of cells, that of ectoderm, mesoderm and endoderm. So they have done it by retroviral mechanisms. They have used OCT4, SOX2 and KLF. I'm not sure why CMYC was not used, or probably it wasn't required for some reasons. So using these three, by overexpressing OCT4, SOX2 and KLF in these through retroviral vectors, they could isolate, they could get induced pluripotent stem cells. The tail tip fibroblasts are completely differentiated cells. Now induced pluripotent stem cells are de-differentiated. That means they do not have any function other than to give rise to different types of cells. So into these induced pluripotent stem cells, they have put in the sleeping beauty transposon with the micro utrophin, and in combination with the GFP with an internal ribosome entry site. So this is the actual repair part. So they have repaired, or they call it as corrected induced pluripotent stem cells. They would have isolated for those that were producing GFP plus. If cells are producing GFP, that means those cells must have integrated the transposon. And using FACS in the last class, I think we have discussed about FACS too. So using FACS, they could separate what are GFP plus and GFP minus cells. Then they have put in PAX3. Now we need to develop the muscle tissue. So they have used PAX3, which is a kind of marker for towards the driving the cells towards the lineage of muscles. And then they developed, they used embryonic bodies in which they have expressed, they have had PAX3 with an inducible promoter. Inducible promoter, if you by doxycycline, once you add doxycycline, it will produce a lot of it, and then eventually the cells will differentiate into the muscle cells, myogenic precursors. So again, using FACS, they were able to segregate them, and then they got the corrected myogenic precursors, and then they transplanted back into certain tissues, skeletal muscles, leg and so on. So when they did, they performed, and there are so many results that were shown, but we are not going into all the details, but I hope you got the point of it. First step is to make the induced pluripotent stem cells. Second step is to make the repair, genetic repair, using micro utrophin transgene. The third one is to develop or generate the myogenic progenitors, and fourth is to transplant. And then they have, these are all experiments to show that this is what happens and so on. So in the control IPS, you see that there is no GFP, whereas in the utrophin, where it was integrated, you can see that there is a lot of GFP, and then they will take those cells and do it. Within the uncorrected one, you do not have cells that are not found here, whereas in the corrected one, where GFP is produced, you will see this part like this. Those are the, I hope you can recollect about FACS, and that is what it is showing. Okay. Here is they are going to, you already must be knowing qPCR, quantitative PCR, or real-time PCR. They were trying to show, prove that utrophin gene is produced and so on, or mRNA utrophin is expressed. UNC is the uncontrolled one. There is nothing here and there is nothing here, whereas during the stages of proliferation and at the later stages of differentiation, there is high expression of micro utrophin. The same thing this is showing that the mRNA is present, of utrophin is present, but it doesn't mean that there is the protein is also present. So to show that, they have used Western blot or immunoblotting to show that this is the control, uncorrected one, and here is the proliferation stage, and this is the differentiation stage. You can see that there is high expression of utrophin protein, right? They've also done several other experiments to show that the integrated or the repaired or integrated transgene, that is micro-utrophin, actually gives the function of the cells. What they have, they integrated it, and then they could show that the mice were able to perform certain functions, and that is how as a proof of concept, this is how it is going to be. I think this is a good start, and people have done again, you will use a lot of experiments with something like this, with the functional, this is the functional improvement and satellite engraftment of transplanted muscles. There are many other fluorescence microscopy and others. I have skipped them. I hope you get the point that these mice are able to recover to a large extent many of the functions, and that is why it is a proof of principle, and it is in 2013. Probably in the future they will be able to do many more experiments to find a solution. So I want you to get the essence of what is there in this slide, right? This is making induced pluripotent stem cells, gene therapy, differentiation, and transplantation. That's the cycle of ex vivo gene therapy in many cases. Okay, if you have any questions, you can please contact, but otherwise I'll try to post the paper, or you can, it's freely available, try to have a look at it. Thank you very much. Bye bye, stay safe.